So, what makes you, well, you? When you get right down to it, life isn't some kind of magic. It's actually a set of really elegant rules. In this explainer, we're going to unlock that code of life, diving right into the core principles of biochemistry that run every single living thing on this planet. It really all boils down to this one huge question. Just look around you. Life creates these incredibly complex, ordered things, like us, out of the total randomness of the universe. How in the world does that happen? Well, that's the puzzle we're going to solve today, by digging into the rules that make it all work. Okay, so before you can build anything, you got to talk about the budget, right? In biology, every single action, every thought, every muscle twitch, it all has a price tag. And that price is paid in energy. So let's start there, with life's energy currency. The big concept you need to know here is called free energy, or as you'll see it, delta G. The easiest way to think about it is like the ultimate decider. It tells you whether a biological reaction can even happen on its own. It's basically the amount of useful energy a system has to actually get something done. This one value tells you almost everything. And this brings us to a really fundamental rule. If a reaction has a negative delta G, it's spontaneous. It just happens. It releases energy, kind of like a ball rolling downhill. But, and this is a big but, if it has a positive delta G, it is not spontaneous. It needs energy put in to make it go. That's like pushing the ball back up the hill. Building things, thinking, moving, pretty much all the cool stuff life does requires that kind of energy put. So how does life pay for all of this work? How does it push all those balls up the hill? It uses a universal molecular currency, and its name is ATP. Honestly, you can just think of it as a tiny rechargeable battery. Cells charge it up, and then they spend it wherever energy is needed to make something happen that wouldn't happen otherwise. And we're not talking about pocket change here. When you break one of those high-energy bonds in an ATP molecule, it releases a really significant chunk of energy. It's just enough to power other reactions and basically pay off their energy debts. It's like the perfect $20 bill for getting specific jobs done inside the cell. Okay, so we've got our power source, we've got our charged-up batteries, but how does life actually use that energy to build stuff? What's the invisible glue that holds all the molecules of life together in their specific functional shapes? You might be surprised to hear it's not some kind of super glue holding it all together. Instead, life depends on a whole bunch of weak interactions. Yeah, weak. By themselves, they don't amount to much, but when you get them all working together, they are incredibly powerful. We're talking about things like the hydrophobic effect, hydrogen bonds, and a few others. They all team up to sculpt proteins and DNA into their perfect shapes. Now, of all those forces, one is the undisputed heavyweight champion when it comes to building proteins, the hydrophobic effect. The best way to picture it is like a bunch of shy people at a really loud party. The parts of a protein that fear water, the nonpolar parts, they all want to hide. So what do? They all clump together in the middle, away from the water. And that simple action is the main thing that drives the protein to fold up into its compact, functional shape. And this leads us to one of the most amazing processes in all of biology, protein folding. How does a simple, one-dimensional string of amino acids just spontaneously fold itself into a complex, three-dimensional machine that can do a very specific job? It's incredible. It all begins right here, with the primary sequence. That's just the order of amino acids, one after another. Think of it like letters in a word, or words in a sentence. This simple, linear code contains the complete set of instructions for building the final, working protein. Now, here's a really important piece of the puzzle. The bonds that link these amino acids together, the peptide bonds, they're special. They're rigid and they're flat. That means they can't just spin around freely. And that rigidity is key because it massively limits the number of ways the chain can fold, which sort of forces it down a predictable path toward its final shape. It's like building with Legos that can only connect at specific angles. And this all leads to a Nobel Prize-winning discovery by Christian Anfinsen. What he showed was, if you take a perfectly folded protein and use chemicals to unfold it into a straight line, it will spontaneously refold back into its correct active shape all by itself. The takeaway was revolutionary. That simple line of code, the primary sequence, contains all the information needed to create the final 3D structure. The code is the shape. So we have energy and we have structure. Now we need action. We need the molecular workers that can actually do the thousands of chemical reactions happening in a cell every single second. These workers are the enzymes. So how do we know if an enzyme is any good at its job? We basically look at two numbers. First is Km. That tells us how tightly the enzyme grabs onto its target molecule. A low Km is what you want. It means the enzyme has a really strong grip. The second is Kcat, and that's all about speed. How many reactions can it pull off per second? The absolute best enzymes have a low Km and a super high Kcat. They grab on tight and they work incredibly fast. But just as important as making reactions go is knowing how to make them stop. This is absolutely critical for controlling everything in the cell, and it's also the basis for how a ton of common drugs work. There are three main ways to block an enzyme. A competitive inhibitor fights for the exact same spot as the target. A non-competitive one binds somewhere else and just changes the enzyme shape so it doesn't work as well. And then there's the uncompetitive one, which is kind of sneaky. It waits for the target to bind first, and then it swoops in and locks everything in place. Each strategy has a really distinct effect on the enzyme's performance. All right, let's zoom out one last time for the big picture. We've met the workers, the enzymes, but where do they get their instructions? And what powers the entire cellular factory? This is all about connecting the master plan to the power grid. And the master plan, the blueprint for every single protein, every enzyme, every part of the cell is stored right here, in DNA, specifically in this iconic double helix shape. This incredibly stable, elegant molecule holds all of that primary sequence information we talked about earlier for everything the cell will ever need to build. But look, a factory that has a blueprint but no electricity is pretty useless, right? Well, the power grid that generates almost all of the cell's ATP, all those little batteries, is the electron transport chain. This is where the energy from the food we eat gets converted into the chemical energy of ATP. So how does this power grid actually work? Oh, it's just a beautiful process of energy conversion. First, a chain of proteins pumps protons across a membrane, building up this huge gradient. Think of it like water piling up behind a dam. This creates a powerful force. Then, this amazing little molecular turbine called ATP synthase lets the protons flow back through. And as they rush through, they physically spin the machine, which jams the phosphate onto ADP, creating tons and tons of ATP. And there you have it. When you put it all together, you see it's not magic at all. It's a beautiful system of elegant, interconnected rules. The laws of energy tell us what's even possible. A bunch of weak forces work together to build complex structures from a simple code. And an army of enzymes powered by a seriously sophisticated energy grid does all the work. And that leaves us with one last, really important thought. We've just walked through the fundamental rules that keep a healthy cell running. So what does that really mean for us? It means that almost every disease you can think of, from a genetic disorder to a problem with your metabolism, can be traced back to one of these rules being broken. A protein that misfolds, an enzyme that doesn't work right, a power grid that's inefficient. Understanding these rules is the absolute first step to figuring out how to fix things when they go wrong.